Cette conférence va maintenant être enregistrée.
You already introduced my research group a little bit. I would just like to tell you that we just a month ago we rebranded our group name, so we are now the CMOR group, Cancer Imaging and Multiomics Research, and that is because we had to renew ourselves a little bit because previously this group and this our science is based in the MR community and the development of MR methodologies, but in cancer research, as most other fields, you can no longer rely on one technique. Those days are past, and so metabolomics has since the early 2000s been one of our main methodologies, but importantly, this is now combined with other molecular analysis, and we also do imaging. So we have the possibility to span a translational line from cell cultures, tissue, biofluids, animal models, and all the way to clinical studies. Let me see. Yeah, so our vision is improved cancer treatment and patient care. And just to show you a little bit about our infrastructure, I'm just wondering, can you see my mouse when I move it, or is there a trick? Maybe I can. Now you see my laser pointer. Yeah. So this is an overview of our infrastructure. So we maintain several biobanks with tissue and biofluids from cancer patients. This is enabled through our, so the research group itself is university-based, but we have a close collaboration with the hospital, the university hospital, St. Olav's Hospital. And so we maintain these biobanks, and we also have access to relevant animal models. And then we also can take part in clinical trials. And this 3T scanner is owned by the university, and the hospital uses it part-time for their clinical examinations. So the equipment ranges from spectrometers and to animal scanners and to the clinical scanners. And interestingly, we've also, at the start of the COVID, we had an installation of Adopiflex, which is for spatial multi-MSI measurements. And we also have a GCMS in our lab now. But for all the other technologies, we use other core facilities at the universities to do the work. So with the hallmark, so cancer in mind, magnetic resonance has ample opportunities to provide relevant information about all the acquired capabilities of cancer. So tumors, they have a abnormal vasculature, chaotic blood vessels, and this is something we can visualize by dynamic contrast enhanced MRI, for example. We have diffusion-weighted MRI to say something about the cellularity. We can use PET for other molecular information, and then the metabolism, of course, which is then the main scope of this talk. So I will introduce you a little bit to the field of breast cancer, and I will give you some snapshots from our metabolomics research across the cancer timeline. So breast cancer is the most common cancer disease in women in Norway and also across the world. There is an accumulated risk at 80 years of 10.5%. Survival rates are relatively high, but there are also, due to increased diagnostic tools and also increased treatment options, there are many cancer survivors. And so this means that efficient management of diagnosis and treatment has a huge individual, societal, and economical impact. So we need good solutions. Breast cancer is also a very heterogeneous disease, and several subgroups of breast cancer with varying prognosis and treatment strategies can be described at the various omics levels. So on the genomic level, mutations of the tumor suppressor genes, BRCA, causes increased risk for breast cancer. On the transcriptomic level, we have the intrinsic genetic subtypes. On the proteomic level, several subgroups have also been defined. The estrogen receptor status determined by immunohistochemistry is an important prognostic and predictive factor in breast cancer. The prognosis is usually very good for estrogen receptor positive patients, and targeted endocrine treatment can be provided. While the presence of the human epidermal growth factor receptor 2 promotes the growth of tumor cells, but because targeted treatment is available, the prognosis is now actually quite good, also for the HER2 positive patients. Triple negative breast cancer is a challenging subgroup with relatively poor prognosis. Currently, no targeted treatment is available, and the patients are mainly restricted to chemotherapy. Yes, and I will, in this lecture, then try to go a bit more into the characteristics of metabolic subgroups of breast cancer. But cancer systemic treatments have really, well, breast cancer treatment is multimodal, but the various systemic treatments have emerged as a great weapon against aggressive and highly prevalent disease, which has led to improved quality of life and preventing millions of deaths worldwide. However, these treatments come with some side effects. Fatigue, cognitive function, metabolic effects, and cardiovascular risk are especially recognized as common side effects of the breast cancer treatment. And so there, with this in background, then several important research questions arise. So how can we do better in treatment stratification? How can we measure who responds to the treatment? And how can we better identify who will experience side effects? Maybe they need additional follow-up, and we need to help them back to the normal life, back to the working life. And who needs this additional treatment and follow-up? Who has the worst prognosis? So I will not say much about metabolomics. This is a field I'm sure you are all well acquainted with. But in cancer cells, it's important to remember that they possess distinct metabolic characteristics, and they change during disease progression and treatment. And so metabolomics has been extensively applied for research in cancer detection, characterization, prognostication, and treatment monitoring. And where is this relevant in breast cancer? And metabolomics can be applied for applications along the entire timeline of cancer, and by that potentially inform the diagnosis and treatment monitoring by detection of prognostic and predictive biomarkers. But I think it's also important to remember that metabolomics can also aid the mechanistic understanding of the molecular changes occurring in cancer progression and treatment, and can also potentially identify targets for metabolic inhibitors. So, where to start? One can analyze the tissue samples from the tumor directly, or one can choose to analyze biofluids. And in this talk, I will only focus on studies where we worked with samples from human breast cancer through the biobanks we have access to. So Our methods. NMR is a simple and robust and by nature quantitative method. And this is what we like to say to those people that mainly work with mass spectrometry. For the analysis, if we start with the biofluids, the biofluids are just mixed with a buffer, transferred to the NMR tubes, and then ready for analysis by NMR. So for the biofluids, we have implemented the in vitro diagnostic protocols established by Bruker, the IVDR protocols, if you've heard about it. This standardized analytical scheme enables detection and quantification of numerous lipoprotein subclasses and small molecular metabolites. And the analysis and quantification is run in complete automation. So this is, of course, a very easy way to analyze large cohorts of biofluids. I will not talk so much about urine analysis today. I will mainly focus on what we can achieve by analyzing the lipoprotein subclasses in serum. But I will also talk a little bit about tumor tissue, as this high-resolution magic angle spinning NMR spectroscopy method is what is really where we started in Trondheim, our work with breast cancer. And regarding this method, we can analyze the intact tissue. And the resolution is approximately similar to what you would see if you do extracts of the tissue and then do ordinary liquid NMR analysis. So we just cut the tissue in appropriate pieces, transfer it to such rotors, as you can see here, with a small amount of buffer containing heavy water. And then high-resolution NMR spectra are acquired through this magic angle spinning method, which I find the name a little bit misleading, calling it magic, because this is pure physics. But the sample is spun at high speed here at the magic angle, and this results in these highly resolved spectra. And for the acquisition, we have implemented very similar acquisition protocols as those that are standardized for serum measurements. So this is a typical HR mass spectrum from a breast cancer tissue specimen. Yeah, what you see here? Well, there are not a high amount of metabolites compared to what you can obtain, of course, with the newer mass spectrometry methodologies out there. However, the metabolites of the metabolome are densely interconnected, so that a modification in any pathway is likely to alter the concentrations of some of those metabolites that we can measure by NMR. And the resulting fingerprint includes information from several pathways known to be affected by cancer. And one of these most studied pathways is the choline metabolism. Phosphatidylcholine, which is the most abundant cell membrane constituent in mammalian cells, and increased levels of choline-containing compounds, such as the free choline, phosphocholine, and the glycerophosphcholine, are detected in several cancers and are considered to be associated with a malignant progression. Normal cells extract energy from glucose, which is converted to carbon dioxide by mitochondria in the presence of oxygen. But if the oxygen supply becomes inadequate, the cells produce lactate instead. And in cancer, glucose is converted to lactate even in the presence of oxygen in a process called the Warburg effect. So altered glucose metabolism has two important consequences. It conserves carbon for the synthesis of macromolecules, and second, it results in a local acidosis due to excessive lactate production. But cancer cells not only depend on increased glycolysis, but also require continued mitochondrial function for the metabolism of glutamine. And glutamine metabolism serves as an alternative source of energy and is important for maintenance of the cell redox balance. And several cancers are described to be glutamine addicted. So to try to sum up a little bit here, so the tissue analysis provided by NMR provides a signature of metabolites from cancer-relevant pathways. An important feature of this magic angle spinning technology is that the sample remains intact after analysis. So you can enable subsequent analysis from the exactly same tissue piece. That means you can extract RNA with high quality DNA. You can do histopathology, extract proteins. So you really have a possibility to do analysis on various platforms with the exactly same sample. Also, what is interesting is that you have this translational potential. We have done work, this is from prostate cancer, where we have shown that there is a high spatial matching between what you see ex vivo and what you see in vivo when you do in vivo MR spectroscopy. But one important thing to remember is that magic angle spinning will always be a volumetric measure. You have no, and the samples are heterogeneous. So, so what you obtain is a bulk measurement. And this is something that can be improved by looking into other and newer technologies. For example, multi-imaging. I will focus on the NMR approach today. So just to go a little bit into also what we see when we analyze the serum samples. And with the IVDR approaches that we implemented in the lab, we can quantify around 30 small molecular metabolites. And in addition, it's now possible to perform this lipoprotein subclass analysis. So the lipoprotein particles are micellar structures carrying the lipids in serum. And the quantification of these subclasses of these lipoproteins are based on are actually based on these lipid peaks that you see here, and a regression model which is embedded in the post-processing protocol, where the shape of these lipid peaks will reflect the distribution of the lipoprotein subclasses, and that to a much more detailed level than what ordinary clinical chemistry measurements can do. So while standard clinical chemistry measurements will provide the concentrations of triglycerides, LDL, and HDL cholesterol, these protocols will provide concentrations of phospholipids, cholesterol, triglycerides, free cholesterol, and apolipoproteins in many subclasses of these lipoproteins. So a much more fine-tuned picture. Yeah, so if you then compare serum to or look into serum, so serum reflects the entire organism. So and serum metabolite concentrations are tightly regulated by a numerous homeostatic mechanisms in the body. And it may be challenging to relate metabolic profiles directly to cancer cell metabolism. And also because NMR has relatively low sensitivity and potential metabolites excreted by the tumor may not be detectable by NMR. And in a recent study, we measured metabolites in tumor tissue and serum from the same breast cancer patients. And here you can see that there are quite strong correlations between the metabolites in the same matrix. But correlations between serum and plasma serum and tissue is really low. So it's therefore important to remember that what we observe when we measure serum metabolites in cancer patients are more related to these tumor-host interactions. And also the serum metabolites in cancer patients will be influenced by a number of other variables, such as genetics, diet, smoking. So it's a complex picture. But I will now start by talking a little bit about analysis we've done using tissue samples and the magic angle spinning approach. So just to provide a context here, we wanted to look into, can we, in cancer patients, in breast cancer patients who did not have any treatment before, could we identify subgroups of breast cancer? And the background context here is this work performed by Peru and Sörli, and now more than 20 years ago, where they detected the genetic subtypes of breast cancer. And these genetic subtypes, such as the basal-like and the luminal types, they are associated with outcome, where the basal-like subtype has the poorest prognosis. And actually, these subtypes, genetic subtypes, are now introduced in clinical routine for improved risk classification through the PAM50 molecular classification algorithm. But what about the metabolic side? We wanted to try the same top-down approach based on the magic angle spinning analysis of breast cancer tissue. And by exploiting unsupervised hierarchical cluster analysis, we identified 3 naturally occurring metabolic clusters. What was really interesting was that the genetic subtypes that I showed on the previous slide, they were evenly distributed across these 3 clusters, which means that these clusters brings additional information. And they also have very characteristic patterns in here. For example, there is one of the clusters had a elevated choline metabolism. Another one had an elevated lactate and reduced choline metabolism, while the third one here had a higher glucose to lactate ratio, which made us hypothesize that this maybe could be a group with better prognostic outcome. But also this shows that these clusters points to these clusters may also be interesting in terms of treatment stratification to metabolic inhibitors, for example, targeting the choline metabolism and the lactate conversion through lactate dehydrogenase. And these are drugs that are, I don't know what's really happening on that drug development side, but it proves the principle that you could find metabolic traits that you could treat with metabolic targeted drugs. So quite some years after this work was performed, we also received the long-term follow-up data of these patients, and we were actually able to prove that there are differences in survival between these clusters, where this cluster 2 definitely has a better prognosis. So next step in this work now is that we will start working with integrated analysis of all omics levels, and we're planning to investigate this through a multi-omics factor analysis that we are implementing. So metabolic changes during treatment is also interesting because most patients with large breast tumors, they regularly receive neoadjuvant chemotherapy, which is treatment given prior to the chemotherapy to downstage the tumor and to make it operable. In this study, we were interested in investigating the metabolic changes during such treatment. So biopsies were collected before any treatment was given. It was also collected at 12 weeks into treatment, and then at the surgery where the tumor was removed. And you can see here on the spectra on the right-hand side, these are mean spectra, and how they are distributed here according to the trajectory line. You see that the distribution changes here, where this is the time point at surgery. So the metabolic profiles are definitely changed. And in this study, we showed that all patients had a metabolic response to the treatment. And we also found that there was a metabolic switch from low to high glucose to lactate ratio, and from high to low total choline and glycine in good responders. And this kind of shows how MR spectroscopy can complement the elucidation of underlying mechanisms affecting the pathological response to treatment. What's interesting is that this treatment response is not necessarily related to survival. But also here we had later access to 5-year follow-up data. And when looking into the spectral fingerprints 12 weeks into treatment, we found a significant association. You can see the red dots here. It's a small study, not so many patients, but it's significant. And some of the metabolic patterns were also here related to higher glucose and lower lactate levels in those that are good responders or have long-term survival. In this study, we also analyzed the serum samples. And when we analyze those samples, there is no significant association to long-term outcome. And this probably reflects that the tissue measurements are a much more direct measurement of the tumor, while the serum is tightly controlled. Okay, so just a few pros and cons of the magic angle spinning methodology. So it's simple sample preparation, safe metabolite identification, provides a characteristic cancer signature, the sample remains intact, and in vivo translation is possible. However, we do observe relatively few metabolites. Sensitivity is relatively low. Automation is still not implemented. Heterogeneity is not accounted for. And biopsy procedure is invasive and sample material is limited. So one way here to overcome the heterogeneity issue is, of course, to move on to more spatial analysis, such as multi-imaging. I will not talk about that today. I will focus on studies exploiting serum for metabolomics instead, and then the NMR approaches that we have implemented in the lab. So you can ask, is there any relevance of lipoprotein subfraction analysis in breast cancer? And actually there is. Risk factors for cardiovascular disease also increase the risk of breast cancer. Obesity is associated with higher risk for more and advanced disease stage and higher cancer mortality rates. Chemotherapy is associated with weight gain and body fat increase, which may increase recurrence risk and decrease overall survival. So definitely, this lipoprotein subclass analysis by NMR is a relevant tool in this context. So with this, we wanted to start out early in this timeline of cancer here to identify possible associations between lipoprotein subfractions and long-term breast cancer risk. So to do this, we had access to samples from a large population study performed in mid-Norway. Half of the participants were women. They were at that time healthy, and these samples were collected in the mid-90s and stored in a biobank, and with a associated data bank alongside. So in this cohort, we coupled this register to the Cancer Register of Norway and identified approximately 1,300 patients, or at that time healthy women, but that later developed breast cancer. And then we also picked out 1,300 matched controls according to a nested case control design. And then from the biobank, we obtained a lot of lifestyle factors from the surveys being done. And from the cancer registry, we got more detailed information on the cancer. And then we used the broker IVDR protocols to identify serum metabolites and lipoprotein subfractions. And so I will come back to that. But one thing that became clear to us when we started working with this cohort was that the samples, which had been stored since mid-90s, they had actually been thawed and frozen several times, and the biobank couldn't. This was in the early days of biobanks, and they didn't really have good statistics for which samples and how this had been done. So we designed a study to look into this, how repeated freeze-thaw impacts the measurements. So we collected serum samples from 20 healthy subjects. These were aliquoted to 5 aliquots each and were subject to a different number of freeze and thaw cycles. So first one frozen once, 2, 3, 4, and 5 times. And then we did the NMR measurements. So this shows this figure shows this shows data from 2 patients, but all measurements, you can see it's more lines here, but they are pretty close to each other. So so you can see here visually it doesn't seem like the freeze and thaw process impacts much. And actually, when we did statistics on this, we found that the median coefficient of variance for the lipoprotein subfraction measurements were only 4.3%. We also did this for urine, and for urine, where we quantified metabolites, and we also quantified the serum metabolites, and you can see that it's consistently higher for the serum metabolites. And that's also why we normally don't use the IVDR automatically quantified serum metabolites, but rather use our own scripts for that. But for the lipoprotein subfraction measurements, it shows that NMR is robust, reproducible, and only minimally affected by up to 5 freeze-thaw cycles. So then maybe we could go back to the biobank analysis. But then we also realized that these samples were collected not at the hospital, but out in buses and cars in the districts. So also there was a variance in how long the blood samples were stored before they were centrifuged. So we were back to the methodologies again to investigate how a centrifugation delay would impact the analysis. Also here we had 20 anonymous donors, And we introduced 30 minutes was what was the gold standard, which is a clinical recommendation, and then we delayed to 1 hour, 2 hours, 4 hours, and 8 hours. And you can see here, because these are average raw serum NOSI spectra, including samples with different delay in centrifugation. And you can see there are large differences in the concentrations. In specific, lactate is increasing, glucose is decreasing, while valine here, this example, is pretty much stable. And if we look at the principal component analysis, we can see that there are large and systematic changes in the metabolic concentration that depends on this centrifugation delay. However, this is quite different for the plasma lipoproteins, where you actually see that the between patient variation is much larger than what the different centrifugation delays causes. So also here we found that the lipoprotein subfraction measurement by NMR is robust, reproducible, and largely unaffected by delays in centrifugation. So then back to the Hunt cohort and the risk prediction. So when we started with this cohort, one of the first things we observed was that postmenopausal women had significantly different lipid profiles compared to premenopausal women, with elevated levels of most lipoprotein subfractions, except for HDL3 and HDL4 cholesterol and phospholipids. So that meant that for the further statistical analysis, we had to focus on pre- and postmenopausal women separately. So menopause causes increased blood lipid levels. Yeah. And to test for the associations of the lipoprotein subfractions and long-term risk of developing breast cancer, we applied unconditional logistic regression. The model was adjusted for confounding factors such as age, number of full-term pregnancies, age of menopause, BMI, alcohol consumption. And for the premenopausal women, we found a significant inverse association with, so these are here, with long-term breast cancer risk. And all of the lipoprotein subfractions with a significant association were VLDL subfractions, except for HDL4 triglycerides. And all associations were in the inverse direction, with odds ratios ranging from 0.77 to 0.83. Actually, in this cohort, we found no significant associations to breast cancer risk in these postmenopausal women. So, what is the biological implications of these results? Well, lipoprotein metabolism consists of two main pathways, the exogenous and endogenous pathway. Dietary lipids are transported through the exogenous pathway, while the endogenous pathway starts by synthesis of VLDL by the liver. VLDLs are large particles consisting mainly of triglycerides. They are produced in the liver and take part in the endogenous path, where they are transported to the bloodstream and deliver free fatty acids to the peripheral tissues and muscles. During this process, VLDLs are reduced to IDLs and finally to LDLs as fatty acids are cleaved off. We believe that these found associations reflect some sort of impaired estrogen synthesis, as impaired estrogen signaling has been shown to be associated with the development of estrogen deficiency, which in turn causes accumulation of triglycerides and increased VLDL production in the liver. And so this is also interesting because we have now taken up to establish a LCMS approach to measure steroid hormones in serum for breast cancer patients. So the next step here is also to make a deep dive in lipids using MS-based lipidomics, and that's being done with using a commercial Biocratis kit. Yes, I don't know how is my I can't actually see the time. Do I have I spent my time, or how is it? How's that? Okay, I'll just keep it going then. I don't think I have so much left. In a different study, we aimed to characterize changes in lipoproteins and circulating metabolites with different treatment modalities. And you can see the design of the study here. So the patients went in for surgery, and the samples we obtained were obtained at time points after surgery. And we had several treatment groups here, which you can see the definitions of here. Some had very combined treatment, others only had surgery and radiotherapy. But we also wanted to compare the longitudinal changes here in 10-year survivors and non-year survivors. So here we're talking, I don't remember the time points here, but 6 months after surgery, and then years here and 7 years at T5. That's what we have. No, this is 1 year. T5 is 1 year, and so these are only months apart. And to exploit this data, we used a repeated measures ASCA Plus for analysis of longitudinal intervention studies with multivariate outcome data. And Associate Professor Guro Giske Ødegaard is really the one who's implemented a lot of our bioinformatic.
Sorry, Tone. Can you conclude your talk?
Yes, okay. Absolutely. I tried to ask if I.
Oh, sorry. Sorry, no problem.
I can't see the watch here. No, I will definitely conclude. So I can say that we found that treatment induced long-term changes into a more heterogenic lipid profile. There were differences, although not persisting throughout here, whether the radiation therapy was local or local regional. And we also found that the non-survivors had different profiles already at baseline, and this persisted the first year after treatment. So to summarize, MR-based metabolomics is a versatile tool in cancer research. Numerous studies show promising results in all parts of the cancer timeline. However, the clinical implementation is still low, and I think that is something we can do something about. We need to standardize, we need to validate, and we need to ensure open access so we can compare and validate each other's data. And we need to remember that metabolomics is a cross-disciplinary field. So this collaboration between scientists and physicians is really important. So thank you very much. And I'm sorry I went over time.
Okay. Thank you for this very, very interesting talk, and sorry for interrupt you because we have
you have
other
time. Yeah.
Yeah. Is there some question where we have just time for 1 or 2 small questions? Otherwise, I have one question. I'm wondering of the stability of the lipoproteins in serum, because we also work with with serum in NMR, and when you prepare the sample and you freeze the sample, and after you defreeze this sample and you remeasure the same sample, you have you have a huge differences in the lipoclean. So it seems that in intact serum, it was very stable.
Yeah, this finding also surprised us a bit, but it's really consistent in our studies that the lipoprotein subfractions that we can measure, they are stable towards both freeze and thaw repeatedly, and also for this centrifugation delays. So we think it's a relatively robust and stable method.
It's quite good news. So no, it's quite good news. So sorry, we have no other questions. So I want to. I hope we will have more time to discuss in other meetings, because it was very interesting research, and I thank you again for presenting us some application specifically in NMR and imaging. So we'll continue the program with the young researchers. So many thanks, Tone, for this presentation.
Thank you.
Thank you again, Tone. Now we will welcome Clément Caffaratti, a PhD student from Grenoble. Actually, if you were at Analytics this year, you were lucky to listen his amazing Flash communication, for which he was rewarded with the Flash Presentation Prize. And today, he will give us a talk about metabolic engineering of probiotics to enhance immuno-metabolic production. So yeah, I think you can share your screen now, Clément.
Okay, is it working?
Yeah, good.
Okay. So maybe I can start. Okay.
Yeah.
So hello everyone. It's a real pleasure for me to talk today. I'm going to talk about my PhD project and talk about the dialogue between the gut microbiota and the immune system. Yeah.
Excuse me, Clément, I think we didn't see your presentation. We see your screen with the cells.
Yeah, sorry. I have to change the slide.
Yeah, maybe.
Like that. Sorry. Let's a second. Okay. Is it working?
Yeah, perfect.
Okay. And so the dialogue between the gut microbiota and the immune system, and I will show you how I use mass spectrometry to follow the metabolic engineering of the probiotics E. coli Nissle. So I think you may know that you have trillions of microorganisms that live in your gut, and those microorganisms are producing a very large diversity of metabolites that can be either directly absorbed by the intestine or reach the bloodstream. And now we admit that those metabolites are regulating your physiology. So in my case, I try to study more the link between microbiota-derived metabolites and the regulation of immune function. And to give you general pictures, we can say that some metabolites have anti-inflammatory properties, such as the short-chain fatty acids, while some others have pro-inflammatory properties. So depending on their nature, they can either induce inflammation or maintain the tolerance in the gut. And if you are interested by this topic, we wrote a literature review on this subject, so feel free to find the review. So in my case, the aim of my project was to understand if it was possible to modify the anti-inflammatory properties of probiotics by only affecting the production of a key metabolite. So for that, we selected the probiotics E. coli Nissle. It is found on the market with the name Mutaflor, and it is already recognized as interesting probiotics because it displayed anti-inflammatory properties. It was evaluated in different mice models of colitis and also in humans. And as a metabolite of interest, we selected spermidine. It belongs to the family polyamine. It is mainly described in human health for its effect on aging. And you have to know that even if your cells are producing spermidine, your spermidine source relies mainly from your diet or from your microbiota if we talk about the amount of spermidine that is found in the large intestine, in the lumen of the large intestine. So spermidine is super interesting because it promotes the differentiation of regulatory T cells in the colon, and also it has some pro-proliferative effect on colonocytes. So it supports both an anti-inflammatory environment in your gut and the renewal of the colon. And also we know that spermidine supplementation protects mice against intestinal inflammation in different models of colitis. So my aim was to produce spermidine in E. coli Nissle and see if it has an impact on its anti-inflammatory properties. So polyamine are a family of metabolites that is shared among, I think, all organisms. And it is also the case for E. coli, so it has its own metabolic pathway for spermidine. So if you are E. coli and you want to produce spermidine, you will first need to get putrescine, its substrate. Putrescine is synthesized in E. coli with either arginine or ornithine. And once the bacteria is able to synthesize putrescine, it can produce a spermidine in a two-step reaction using the SPD and the SPE enzyme. So our first strategy to increase the bioproduction of spermidine in Nissle was to build a synthetic biological circuit where we place the SPD and the SPE gene under the control of a constitutive promoter. So it was the construction PCC25. And to study if this strategy has an impact on spermidine production, we perform liquid culture and we collect the supernatant of culture at the beginning of the culture and 24 hours after the beginning of the culture. And each supernatant were prepared and analyzed using an HCMS approach. And it was used to follow the spermidine concentration in the supernatant and also polyamine-related metabolites. So here you have a brief description of the growth condition. As you can see, the engineering has no impact on the growth rate of the bacteria. And if we look at the spermidine and putrescine levels, so spermidine the product and putrescine the substrate, you can see that the overexpression of SPD and SPE was already a good strategy to increase the amount of spermidine found at 24 hours in the supernatant of the cells. So it was a very good start for us. But when we took the time to look at polyamine-related metabolites, we found that this increase of spermidine was also linked with an increase of acetylated form of spermidine, such as acetylspermidine and diacetylspermidine. So it was a major drawback for us because it means that we were losing a certain proportion of spermidine. So we had to find a solution. So we decided to look at, of course, the metabolic pathways, but also the biology of polyamine in E. coli and the transporter system in E. coli. And we learned that polyamine is super important for the bacteria to maintain its physiology, and also that spermidine accumulation is toxic for the cells. So E. coli has different strategy to escape from this toxicity. And one strategy for E. coli is to overexpress the SPG gene that helps to convert spermidine to the acetylated form of spermidine. So what we saw on the last slide was just finally the normal response of E. coli against spermidine accumulation. And now if we look at the transporter, you can see that you have different transporter in E. coli that help to either import or export polyamine. And one transporter is a spermidine exporter. It is called MDTIG exporter. And we hypothesize that maybe by increasing the MDTIG expression, this will maybe reduce the amount of acetylated form of spermidine. So to test this hypothesis, we built a second synthetic biological circuit where we place a first circuit to increase the biosynthesis with the SPD and SPA gene, and a second one to increase the expression of the exporter, spermidine exporter. So we perform exactly the same type of experiment, the growth rate and the evaluation of spermidine, putrescine, and arginine levels. And as you can see on the spermidine graph, you have a strong increase of spermidine concentration in the supernatant of the bacteria at 24 hours for the PCC35 groups. So it was a fantastic result for us. And now if you look at the acetylated form of spermidine, you can see that this strategy was maybe a very good strategy to limit the amount of acetylated form of spermidine in the supernatant of the cells. So now we have our probiotics that is engineered to overproduce and also export spermidine. But and here you have a general strategy if you want to produce a metabolite of interest in a bacteria and test if a genetic engineering strategy is efficient to overproduce your metabolites. But I think you all agree that it doesn't give you any kind of information about the effect of the probiotics. So for that, you can do in vitro or in vivo models. In our case, we choose the in vivo studies. We use healthy mice to address this question. They receive a daily oral gavage with one of the following treatments. It was either PBS, the wild-type Nissle transformed with a PCC31 plasmid. I'm going to introduce this constriction, or a pure spermidine solution. And then after 14 days of gavage, we use the colon to perform immunophenotyping and study the function and proportion of T helper lymphocytes. So just for you, the PC31 plasmid, you have all the genes that have been already mentioned, but I have just replaced the first promoter that controls the expression of SPD and SPE. In this contrition, we use an anaerobic inducible promoter to be sure that we will maintain a certain level of expression in the gut, and we play with the amount of oxygen that is available in the gut, because the gut environment is mainly anaerobic. So if you use a PFN-RAS promoter, you are going to induce this promoter in an anaerobic environment. So and we wanted to maximize our change to see something. So now if we move on the in vivo results. So here you have a different graph that represent the T helper lymphocytes with that produce different cytokine. So either IL-10, that is an anti-inflammatory cytokine, IL-17, it's a pro-inflammatory cytokine that drive antibacterial responses in the gut, and tumor necrosis factor. And you can see with on the IL-10 graph that the PCC31 groups the mice have an increase of IL-10 producing T lymphocytes. So it means that we are increasing the frequency of T helper lymphocytes that have anti-inflammatory properties. And also for IL-17 and TNF alpha, we are totally decreasing the natural pro-inflammatory effect of the bacteria, so of Nissle, in a healthy colon. So it was a very nice result. It suggests that by only increasing the production of a key metabolite, this engineering totally changed the reaction of the immune system. So it's a preliminary result, and we are currently assessing the role of our engineered probiotics in a mouse model of colitis to see if it can help to reduce the severity of the colitis. So if you have to keep some take-home messages, I think now you know that the microbiota is involved in a constant dialogue with the host immune system. You also know that metabolites that are produced by the microbiota seems to be essential to control immune function in the gut, and also that engineering probiotic strategy can be used to understand either host-microbe interaction or to propose new therapeutic strategy that can be used in different contexts, such as inflammatory bowel disease, for example. So thank you for your attention. I would like to thank the RFMF for giving me the chance to talk today, my research team, my supervisor, and also our collaborators in Toulouse. Thank you so much.
Thank you, Clément, for this really great talk. If you have questions to ask to Clément, feel free to send them by the chat, and we will ask them to Clément. And I think Pascal has already a little question for you, Clément.
Yes, thank you for the presentation, which is very interesting. So I was asking me why you are not measuring spermidine, for example, in stools of the mice, and so you have collected the gut samples. Why not measuring this in the stools?
So we try to measure the amount of spermidine or polyamine-related metabolite in feces, also in the cecum, or in the intracellular compartment of colonocytes. And it was really hard to, with a small group of mice, like 10 mice, to see something, even in the group where we treat the mice with spermidine. So we don't have the right way to measure this concentration.
You identify spermidine in your sample, but there's no difference between the groups. That's correct?
Yeah, it's really variable between the group, even when we give spermidine.
Is it due to absorption of the spermidine?
I think, yeah, I think, and there is a group in Japan that try to measure an information like that using GCMS, but and it seems that it gives better results. But in our case, with our equipment and samples, we were not able to see any kind of difference. And also that you have to know that polyamine, it's a metabolic pathway that is shared with the host. So you have absorption, but also you have degradation, transport, different things. It's super complex to study the evolution of only one metabolite, class of metabolites. In our case, if it was a restricted, microbiota metabolite, maybe it's much more simple. But in our case, as it is shared with the host, it's more complex.
Okay, thank you.
Thank you again, Clément. Now we'll give the floor to Anaïs Draguet. Anaïs is a PhD student from the University of Mons in Belgium, and she will speak us about NMR metabolics in the context of breast cancer. So the floor is yours, Anaïs. I think you can share your screen now. Yeah, perfect.
Yes. So good afternoon, everybody. I'm very happy to present to you my thesis project today, and thank you so much for this opportunity. So I'm working on an ABC transporter called ABCB5, and this one is potentially implicated in metabolism. And I'm trying to prove this hypothesis thanks to the NMR technique. So, of course, I will start with an introduction. ABC transporter are membrane protein, and this protein is integral. Their role are molecule transport, either the import or export. The substrate of ABC transporter are really numerous and belong to many classes of molecules like amino acid, lipids, metals, drugs, and many more. So they are present on cell membrane or organelle membrane. And in the human genome, there are 48 members, and they are divided in 7 families from A to J. So about structure of this transporter, ABC transporter show a particular structure. They have a first domain, the transmembrane domain. The role of this domain is the substrate recognition and the translocation on the other side of the membrane. It's a variable domain. The second is the ATP binding domain. The role of this one is, of course, binding and hydrolysis of ATP. So a complete transporter has 2 transmembrane domain and 2 ATP binding domain, contrary to a half transporter who has 1 transmembrane domain and 1 ATP binding domain. Half transporter could homodimerize or heterodimerize to construct a complete transporter. So about the function of this transporter, the substrate go in the transporter, and this action induces a modification of the transporter conformation. This new conformation exits the substrate on the other side of the membrane. And during the conformation change, 2 molecules of ATP bind the ATP binding domain, and the hydrolysis of both molecules give at the transporter its original conformation. Generally, ABC transporter in prokaryotes are implicated in import and export, while ABC transporter of eukaryotes are implicated in export only. So my thesis is about ABCB5. So it's an ABC transporter in the B family. We found this protein in pigmented cells, so typically melanocytes, and in testicular and mammary tissues. It's a marker of cancer stem cells, sorry, and particularly in melanoma. ABCB5 take part in the drug resistances, for example, doxorubicin or paclitaxel. And there is 3 known isoforms of ABCB5, alpha, which is a little protein, beta, which is a half transporter, and 4-length, which is a complete transporter. So the functional physiological function of ABCB5 is the regulation of progenitor cells, but it's only known at this moment. So the goal of my project is divided in 2 parts. The first concerns the implication of ABCB5 in the development of melanoma. The hypothesis is that the substrate of ABCB5 accumulates in the cell when ABCB5 is mutated, and this accumulation induces the cancer development. I will try to discover the substrate and also the mechanisms. The second concerns the drug resistance. So I will try to see ABCB5 like a potential target for future therapy. So ABCB5 was possible for the export of therapy, and if we block the ABCB5, the therapy stay in the cells. Yes, I will present now my material and method part. So I worked on mice, and more specifically on mice urines. So my mice stay in a metabolic cage during 24 hours. This cage is composed of a circular closed cabin where the mice is placed. In this cabin, there are a food place and a drink water place. Under the paws of mice, we found a brake platform which allows the recuperation of body waste. Thus, we found in the bottom of the cage a collector tube for feces and a collector tube for urines. So after the day in the metabolic cage, the urines are collected and centrifuged. After the centrifugation, the urines are prepared with phosphate buffer to standardize the pH and TSP, or internal reference for NMR. The samples are finally analyzed thanks to an NMR spectrometer with a magnetic field of 600 MeV. So I use two types of mice: wild-type mice, which express ABCB5, and knockout mice, which don't express ABCB5. For the interpretation of the result, I started with a spectral correction with baseline and unfast correction. After that, I carried out a binning. The spectra is divided in region of 0.05 ppm. The area under the curve is measured for each region, and the result of this measure is transferred in Excel file. On this file, I proceeded to a normalization by total area. And finally, I loaded the results on Simca software to perform multivariate analysis, and I identified the discriminant metabolite. So about results, the result of this study show a very good separation of the both group. So this graph is the graph of PLSDA, so a supervised analysis, but the non-supervised analysis show the same separation, so the PCA. These results show that the presence of ABCB5 induces a difference in the metabolic signature in mice urines. So there is the loading plot, so associated at the score plot just before, and it shows the discriminant zone and thus the discriminant metabolite between the both groups. After identification of this discriminant metabolite, we found the metabolic signature in urine of the presence of ABCB5. So you can see that there is a significant difference between both groups in the concentration of isoleucine, leucine, valine, acetylglucosamine, butyrate, succinate, citrate, and oxobutyrate, and dimethylamine, trimethylamine, TMAO, taurine, and creatine are also discriminant metabolites, but the difference between the group is not statistically significant. So in conclusion, the metabolic signature in mice urine highlight two hypotheses about ABCB5 knockout mice. The first hypothesis is cardiac failure. A PhD student in our lab showed a very few years ago a similar metabolic signature in rat urines, and rats have received a cardiotoxic molecule just before. So there are also some other evidences in literature that the cardiac problem show a similar metabolic modification that our mice. So the second hypothesis is type 2 diabetes. A paper of this year showed a comparative study with rat, mice, and human with diabetes. So the pathology was induced for rodents. The study showed a lot of similarity between their metabolic signature and our metabolic signature in urines. And of course, this hypothesis is confirmed, but with other references. So I don't know yet which hypothesis is confirmed, but both could be linked to. And to go further in this study, I plan to work on other models like cells, of course, but also on chicken embryo. I plan also to try the detection of cardiac and diabetes markers in urines and to carry out the same metabolic analysis on serum of mice. Thank you for your attention.
Thank you, Anaïs. So we have two questions in the chat. Mathieu asks you, how do you normalize your urine data?
Sorry, I don't hear you.
Sorry, it's okay now?
Yes, yes.
Okay. So Mathieu asks you, how do you normalize your urine data?
Yes, yes. It's a normalization by total area.
Okay. It's okay for you, Mathieu? I think so. I think yes. And the second question, did you analyze faces?
No, just urine and after the serum.
Okay. Mathieu asks you if there is no difference of concentration between urine samples.
Yes, it's possible, but the normalization by total area did this difference during the analysis.
Okay, thank you. Have you got any other question in the chat? Mathieu, thanks you. Okay, I think it's okay. So thank you, Anaïs, for this great presentation. And now Mathieu will give the right to Tristan. So we will now welcome Tristan Cardon. Okay, it's okay. Tristan, are you here?
Yes, I'm here. I'm trying to connect to be sure that I saw the good things.
Okay, so Tristan is the winner of the poster award at the Analytics Conference in Nantes last month. He is a postdoctoral researcher at the PRISM laboratory in Lille, and he will present his work entitled When Silencing is Noisy, Identification of Alternative Proteins by Proteogenomic Approach. So please be free to ask questions in the chat in French or in English during the presentation. And Tristan, it's yours.
Okay, thanks. Do you listen to me? Yeah, it's okay?
Yeah, it's okay.
Okay, perfect. So many thanks for the invitation first. So I'm working in proteomics, so I hope I will not disturb a lot of people and we will not lose everyone today. But so today I will introduce you of my work about the alternative protein and what we do with alternative protein and what is the impact, what is it an alternative protein in the cells. And how we connect it also now with all the kind of omics. So I'm coming from the laboratory of PRISM in Lille. We are in CERN and we are connecting also by other company. And this work I will present to you today is also in interaction with some team in Italy and in Canada by Xavier Uku or Giorgia Simonetti. So first to recapitulate and to remember you, when we spoke about protein, we spoke about things coming from a gene from the DNA, who is coming from different transcript, who can express one mRNA. On one mRNA, we describe different parts, the 5' UTR, the 3' UTR, who are described to be non-coding parts, and the CDS domain with the start codon and the stop codon. Between that, we will recover the coding sequence for the protein. This coding sequence, since 1999, we described like Cossack sequence before the start codon, will give the best access to the ribosome to fix and to code into protein. This sequence is something used to make also now the prediction of what we call the reference protein. And this will be used also when we do mass spectrometry. Why? Because when we do mass spec in a proteomic approach, the most of the time we will do what we call bottom-up analysis. For that, we will extract the protein from the cells, and from this total protein extraction, we will do the digestion. The digestion gives you the peptide, and we'll analyze the peptide into mass spec. And what we observe is that after the identification based on a comparison with the databases, it stays like more than 10% of the spectrum who are not annotating at the end. That we know now can be also the effect of like post-translational modification, some modification you didn't expect, some modification mutation, for example, inside your protein. But even with that, we didn't explain this amount of spectrum, and we're considering that they have a possibility to lost some information. And this can be due of the formation of the databases we used during the interrogation. And why? Because since we do proteomic analysis in MS, we are using databases to reannotating the peptide we saw. And for making the databases since the beginning of the proteomic, we have to define some rules. And these rules was defined based on the Cossack sequence presence, on the fact that one mRNA code for one protein, and you will keep only the biggest protein coming from an mRNA. And that constructs the first databases we're using. And it was really enough for a long time, and it was enough since now. But we are starting to observe the possibility to have some others production from the same mRNA. And we saw now that you can fix your ribosome inside the 5' UTR, inside the 3' part, or even inside the CDS domain, but with a shift of 1 or 2 nucleotides, giving you another open reading frame, and so another sequence of protein. And that have been described by ribosome profiling and other techniques. And based on this observation, and even in non-coding RNA, we're observing that you could fix some ribosome on the non-coding RNA for the production of new protein not described in the reference proteome and on the databases. And so, based on all the other prediction of possible site of fixation and translation of protein, we are generating a new database called OpenProt. This is free of access inline, and you can see all the rules we follow to make these new databases and to identify new kind of protein and new protein. And so, of course, we are doing also a description of this database and of this new protein, this alternative protein, also called the COS proteome. And what we observe is that this alternative protein have a mean of size of 45 amino acids when normally you have 10-fold more for classic protein, also because of the position inside the mRNA or from the RNA of origin. We observe that also you have a lot of alternative protein coming from the CDS domain with a shift inside the CDS, but also even more, 40% from the 3' part, and few from the non-coding RNA or from the 5' part. So you have a specific repartition depending also of the possibility to fix the ribosome on the mRNA. And what we saw also is the fact that this kind of protein are also conserving inside the different species, and it's something you can recover everywhere. Another interesting fact is that this alternative protein can contain a specific domain with homologue to another protein coding by another mRNA. Even if you identify an alternative protein from gene A with already coding for a reference protein, so you can have a transcript with two different products, the reference protein and the alternative protein, but the alternative protein can have a domain or a function homologue to another protein from another gene, like here, the gene Y. One of these examples is alt-MRV1, an alternative protein coding from the transcript normally coding for MRV1, so 2 different proteins, but alternative MRV1 is also homologous to BRCA1. And that we confirm also by the co-localizations that after the expression of alt MRV1, we identify the same localization almost with BRCA1. So you could play a role as BRCA1, but without exactly the same structure, and so having a double action. Alternative proteins are also protein recovering specific from tumorous tissue or benign tissue. It's something you have to understand that the alternative protein have something from the base of the protein. It's a classic mechanism. It's not specific to the tumors. You can recover everywhere, but they can be also impacted by the mutation or by the pathology. So after observing them, we are also searching for some function of this alternative protein. I will not spend a lot of time on the methodology, but we are using cross-linking to fix the interaction between the alternative protein and the reference protein, and doing mass spectrometry we can identify the reference protein in interaction with the alternative protein and recovering the pathway connecting the alternative protein. And from that, so we are describing some network where we can see some alternative protein involving in the impact of the response to the interleukin, but even more interesting on the translation of the protein. And it was not the first time that we saw alternative protein can be interacting with something from the ribosome or something managing the production of other protein and references protein. Another interesting point for our study in these glioma cells was to observe that some transporter reference protein are connecting to a lot of alternative protein. And from this, we are confirming this interaction observing by cross-linking MS, doing a 3D model of the reconstruction of the protein and trying to dock the alternative protein, the small one, to the big one, the TPM4, a part of the filament protein. And we saw that the alternative protein we are observing by crosslink can be fixed at the position we are observing by the crosslink. And we confirm like that the possibility to transport this alternative protein to be implemented inside the cytoskeleton of the cell. Even to go through this information of cross-linking, we are also doing some confirmation of this interaction by targeting approach. And so for that, we are generating our alternative protein inside the cell, doing co-immunofluorescence to see if TPM4 and alternative protein are combining together inside on the cytoskeleton, what we could confirm here by microscopy. And also we do a specific bioid interactomic analysis where we fix a biotin ligase to the alternative protein, and we are showing all the interactors by the fixation of biotin from the interaction. And here we could confirm that this alternative protein can be fixed with some transmembrane and transporter of protein into the cytoskeleton, but even get an effect on the metabolism of the RNA or to the mRNA function. And through that, we are also searching for alternative protein into sample of patients. And we do, after mass spectrometry imaging, we are doing microextraction in the lab. And from the analysis of all the specific part of the image, we could recover the analysis, by the analysis, the ID of the protein specifically identified in each zone. And from this zone, we could also correlate it with the survival curve of the patient. And what we are observing is that one alternative protein we are identifying in the study is connecting to the survival curve of the patient with glioma. And so now that doing for us a new target to search and to understand into the pathology and to understand as an alternative protein. The bad thing for this alternative protein is the fact that this alternative protein is directly coming from a non-coding RNA, so we have no information about the function or the role of this gene. But the only one things we can get from the genomic part and the phenotypic part is that this gene is something new inside even the human things. It's a gene only coming since the primate and not before. And so it's something only new in the human population. It's the only one paper describing this non-coding RNA connecting to this gene. So we are also doing some focus analysis by bioID to see who are the interactors of this alternative protein into the pathology to recover the different impact and the different signaling pathway involving in this interaction. And in another story, we are working, I am working now in acute myeloid leukemia with a pathology where we get an RFMF project with the goal to make a multi-omic analysis. So from a sample to the patient, and a patient from Sweden, Spain, France, Finland, Germany, or Italy, we get some samples and we will combine. For the moment, it's still in project, but we are doing actually the recruitment of the patient and we are keeping the sample to make the analysis. And we will do the transcriptomic part, the proteomic part, the metabolomic part, and we will combine with all the genome part we get from the patient. And thanks to this collaboration, we structure also another project with the Italian teams, where they, on the same pathologies, they identify non-coding RNA, PVT1, who can be or in 3 different linear form or from a circular form. And this PVT1 has already been described to be involved in the proliferation of some tumoral cell, but we don't know exactly the role into the EML pathology. So they combining 2 types of cells, Kasumi and Oxy cells, in 2 different conditions, the condition of non-oxia or hypoxia, and they inhibited or the circular one only or the linear one only in the 4 conditions. And from this information, we are doing in one part the transcriptomic and the other part the proteomic. And why? We are combining both. The transcriptomic gives us the information of databases we can translate into an in silico translation. The protein we can expect into the proteomic analysis. And from that, we are doing like proteogenomic approach, and we are constructing databases specific for each kind of cells and connecting to the transcript information we can recover. The other interest of that is that in these databases, we will keep also the mutation. And this mutation appears into the DNA, and because of the different conditions, have an impact into the mRNA, but we didn't consider some of this mutation, or we consider it as silencing when, if you observe it into the ORF or the frame of the alternative protein, you can have an impact into this alternative protein. So some mutation describing to be silencing for the reference protein can have an impact for the alternative protein. And it was what we searching and what we wanting to see, the repartition of the mutation for the alternative protein and impacting because of all the modifications they are doing into the cells. So in first, I doing the characterization of the databases we get, because we get several databases for each kind of cell in each condition. And we're observing that we can have referencing protein here in yellow, but also a lot of alternative protein predicted. And the interesting part is here in light color, you can observe that you can have 6% of alternative protein mutated and percentage of protein, referencing protein mutating, and new isoform also. Isoforms they are not considering into the databases, the classic databases. And another things we didn't expect what we observing directly is that in one kind of cells, if you change the condition of culture of of having, you can observe that oxy cells in normaxia, comparing to hypoxia, is predominantly have alternative protein with some mutation. You will increase the mutation of your alternative protein after in a normal condition comparing to the hypoxia condition. Is that something we could also observe in the reference protein, but with the lowest impact? And so we are comparing also the different specific protein, not recovering in all kind of databases. So each cells have this own databases. So you expect also to have some specific protein you will not recover in the other databases. And when we compare this protein from these databases, we are observing that you have also a difference in terms of quantity of alternative protein and mutation into these databases. And the interesting fact is that Kasumi cell, for example, are describing more alternative protein mutation than the reference protein, than the oxy cell, sorry. And with that, we are comparing also the abundances of our protein into this sample. So here I keep you only one part of the study here for the inhibition of the circular PVT1 in two conditions, hypoxia and normaxia. And what we observe that you can recover some specific mutation into that and some specific protein, not mutated, but specific to each condition. And when we observe the protein sharing by all the conditions, we can also see the impact of the abundances of this protein. And we compare the abundances of this sharing protein between the conditions. And what we can observe is that comparing the control without inhibition of circular PVT1 and circular PVT1 inhibition, you have a batch of protein, a group of protein, totally under expressed here after the inhibition, or a group of protein totally overexpressed after the inhibition. It was the same on the hypoxia condition. And so we could also, after correlate this identification protein to the pathways they are involved, to see in which kind of pathway and which biological process or molecular functions they are connecting. And what we saw is that here you have specific protein and specific pathway overexpressed into hypoxia, underexpressed in hypoxia, when here you have pathway more specifically overexpressed in the normaxia condition, and here downregulated in the normaxia condition. As you can see also, you have some pathway in both cases, hypoxia or normaxia, also downregulated. It's also what you can observe here in the normaxia condition. You will also find some little purple with sharing by the hypoxia condition. So, and on that, we are really happy to confirm also the fact that some protein we identify and some pathway we identify are also identified after the analysis of the RNA-seq data they are using, and also the metabolomic analysis they are doing. So we confirm by the proteomics the impact into the same pathways they are observing with other approach and with other kind of techniques. Based on this analysis, we are also comparing the different inhibition of the different form of the non-coding RNA, and what we observe is some group of proteins specifically inhibited in each kind of cells and each condition of harvest. And we can again connect the group of protein to the function and the biological process we know for them. And here, for example, we saw that the metabolite is underexpressed when you inhibit the circular PVT1 into the Kasumi hypoxia. And based on all this identification we get in all kinds of cells, we are connecting to the kind of databases and kind of protein we get into the databases. And what we are observing is that basically you recover the reference protein, but you also have a lot of kind of reference protein with some mutation, specific mutation, and you can get a lot of new isoform, not referencing protein, so also like a ghost proteome of your cells. And from this new isoform, you also have a lot of protein with a mutation into this new isoform. and that is new. And another thing is the fact that some of this sample are presenting a specific alternative protein, few but some. And for the moment, we didn't get alternative protein with specific mutation connecting to the transcriptome. But something also in work actually, because we didn't treat everything for the moment. And what next for the ghost and for the alternative protein now? So into the lab, we are working also now to see if this specific alternative protein can be specifically presenting by the cell or even have a specific localization like at the surface, and that we could use as a new target for new kind of therapy, or like with the CAR T cells, we could target specifically one alternative protein not present in the other, and if they show some specific mutation or things like that. So into glioma cells, we are presenting fact that some alternative protein are recovering specifically at the surface. Now we have to see if they are specific to the glioma, if they are containing specific mutation and things like that. But actually, there are some alternative protein to the membrane. So on that, I would like to thank all people who are working with me, the team in my lab. Here you can see almost everyone into my lab, and everyone has a space into this story also probably. So many thanks also to you for your attention, and if you have questions, yeah.
Thank you, Tristan. If you have any question, you can talk now or write in the chat. Maybe I have one for you. Future cohorts, you are recruiting patients. Have you got any idea of the number of patients you will have and when? In which
kind of, for example, for the email story, we are recruiting actually around 150 sample, so patients from everywhere from Europe. So it's a specific logistic to get all the sample, also because they are taking a specific part of the bone marrow, and it's really difficult to have access to this kind of sample, even more because they are treating also for other kind of analysis, because we are doing the multi-omic analysis on that. So we need a lot of quantity of cells for make the proteomic, but for doing also the metabolomic and the transcriptomic, it's something pretty difficult. Okay. And you are aware with the quenching of the metabolism, for example, the quenching of the samples? Two separate samples for that. Where we are using part of the sample only for the proteomic, and the other part are quenched directly, and we extract the metabolite after the conservation specifically. Okay, thank you. Is there other question? Not in the chat. Okay. So thank you, Tristan, very much for this nice talk. And I will now let Pascal close this nice webinar we had this afternoon with you all. Maybe there is a question. No. Audrey, thanks all the younger speakers and all participants.
Okay, thank you, Audrey, to stay with us until the end, and thank you also for all the participants, presentators, and also people who follow this webinar. So don't forget the next webinar in the 1st of December, and so have a nice evening, and see you next webinar. Goodbye.